Hello everyone, my name is Minh-Nguyet and I'll be presenting to you all today about Legionnaire's disease. So it was first detected after a 1976 epidemic outbreak at the Pennsylvania American Legion Conference, and there were 142 reported cases. And out of those cases, 29 individuals died, which is why it's nicknamed the Philly Killer. It was initially thought that influenza was the main cause of the disease because the symptoms are very similar. However, Dr. Joseph McDade eventually identified the actual cause, which I will discuss in a little bit. Another thing I wanted to note was that it can be traced as far back to sporadic cases in 1959, but the causative agent wasn't identified yet. This is a taxonomy of the causative agent, which is Legionella pneumophila. So the bacteria is the kingdom, phylum is Proteobacteria, class is Gamma bacteria, order is Legionelles, family is Legionelsae, genus is Legionella, and species is Pneumophila. So Legionella does cause two well-known diseases that I wanted to discuss because initially the symptoms do seem similar. The incubation period is far less for the second disease, which is Pontiac fever. Another thing is Pontiac fever is milder in form than Legionnaire's disease. It doesn't cause pneumonia, and it does not require antibiotic treatment. So those are just some of the differences between the two. Specifically, let's discuss about Pneumophila. So Pneumophila is gram-negative bacilli that is Coccobacillus. It's oblique aerobic, and it has fimbriae as well as a single polar flagellum, which you can see here. It's facultative intracellular parasites, which means that wherever it infects or it's growing, it has to grow within another organism. It grows best in 35 to 45 degrees Celsius, so it's pretty hot temperatures, which means that it's hard to kill in just in hot environments. Its pH grows best at 5.8 to 8.5, so our bodies are perfect reservoirs for that because we're at 7.4 pH. Its incubation period is anywhere from 2 to 10 days, and it's found ubiquitously in fresh water. So this includes man-made structures as well as natural structures. I just wanted to note that since it does grow relatively well in warm water, it is harder to kill by superheating. So the pathology, it grows on biofilms within the water where it will multiply in amoebas. And then whenever we receive which is transmission is through aspiration and respiratory droplets. So if we are infected, then it travels to the lungs where it goes to the alveolar macrophages. And there are CD3 receptors that will phagocytize the bacteria and form LCVs, which is Legionella containing vacuoles. And the bacteria will multiply through the T4SS system, which is just a secretion system that makes it hard for lysozymes to detect the vacuole and kill the bacteria and allows it to grow until it's released back into the body to infect other cells. I'd like to note that it is not person-to-person transmittable, which is just what this diagram is showing here. And then this picture shows areas of high risk. So that includes anywhere that there's water sources. So AC cooling towers, AC units, hot tubs, because there's warm water as well. So it's a very good environment. Anywhere that's stagnant water, hospitals, hotels, those are all very high risk places for Legionella to grow. So it is not contagious because it's very rare for it to be person-to-person infected. However, it has happened recently. It's still very new. It is the leading cause of nosocomital community pneumonia, and it is a nationally notifiable disease. So there are anywhere as little as 2 cases within 6 weeks, then it should be reported and prevention measures will be taken to find the source. This is because water can spread the bacteria very quickly and then more cases will occur. So these are just some articles showing the significance of the disease. So this is still occurring this year in October. This is in New York. These are both in New York and Manhattan, so in the US. It's also worldwide, so it can be found in Brescia, Italy. This was in September, and even in Australia, which was also in September. The World Health Organization does report that it's around 15 cases per million individuals not reported. So it's not super high risk, but it can be if preventive measures aren't taken, which I will talk about those measures in a little bit. So symptoms of the disease include headache, high fever, chills, muscle aches, coughs, and this list is very generic, so that's why it's a little bit hard to diagnose Legionnaire's disease initially, because it can be the same as other pneumonia or even influenza. If it progresses far enough, there will be like coughing up of the blood, diarrhea. Eventually, it could lead to other organ failures besides the lungs, and eventually death as well. So to diagnose it, the doctors initially diagnose it with pneumonia, and to further identify what type of pneumonia is or if it's Legionnaire's disease, x-rays are done to identify infiltrates, which you can see here. And there's also a secondary test, either urinary or sputum culture, just to get water or like body fluid from the individual to grow and to identify if there are any antigens in the body fluid that would be positive for Legionnaire's disease. And it's also important to let doctors know if you are getting treated for pneumonia, if you've traveled recently within the last week or been near fresh bodies of water, because the incubation period is only 2 to 10 days, and then symptoms will show. And one thing I forgot to mention was that sometimes it can be slow, so the CDC does say that you should probably even wait up until 14 days and symptoms can show then. So it's very important to pay attention to these things. The immune response is twofold. So there's both intracellular and extracellular. So detection within the cell is by toll-like receptors that will stimulate chemokines, which will stimulate neutrophils to come in and kill the bacteria. And the other one is nod-like receptors, and they will stimulate nods, which is shown here. And they basically cause inflammation within the cell, as well as ISGs, which is interferon stimulating genes. And these interferons will surround vacuoles that have the infectious agent to prevent it from further infecting. The lysosomes can go in and kill them. So together, the natural killer cells, T cells, the interferons, inflammation, neutrophils, all of that usually is enough to kill the bacteria before symptoms occur. So that means that the actual number of infection reported versus how many are actually infected is lower. But sometimes the body is not able to fight it off, and that's when symptoms occur and treatment is needed. So treatment is right now through antibiotics, and it's a number of things. So it can be macrolides, which is azithromycin, quinones like levofloxacin, tetracycline like doxycycline. These are just some of the medicines. There's a lot more. And with treatment, there's a 90% recovery rate within non-immunocompromised individuals, because immunocompromised individuals, it can be as high as 80%. And currently there's no vaccine for the disease. And it has been found that there is a lifelong immunity to the certain strain that you receive. So it's usually serotype 1 of the pneumophila, and so you won't get infected with it again. So risk factors include individuals who are older, chronic lung disease, smoking, immunocompromised or immunosuppressed individuals, underlying illnesses like diabetes or renal failure or hepatic failure. Recent traveling such as exposes the individual to more areas where there could not be good maintenance against the bacteria. Hot tubs, because they're both high temperatures and stagnant water. And healthcare facilities. So the reason healthcare facilities are on the list is because usually it's immunocompromised individuals that are infected, as well as they have high areas of water that flows through, and so it's like a double risk factor. And it's been found that actually up to 7% of the cases reported is from individuals in healthcare facilities, and there's also 20% due to traveling, so it's a very high percentage. So there's no vaccine, so we don't really have that preventative measure, but there are other things that the CDC does have in place, including regular maintenance of cooling towers and water supplies. Kind of making sure that your shower is clean, and you just avoid hot tubs, avoid any stagnant water conditions. Hyperchlorination can kill the bacteria as well as superheating of the water, but there are some complications with that, so you have to be a little bit careful. But in general, as long as it is reported correctly and the areas are kept, such as cooling towers and showers, are kept clean, then we can keep the rate very low. And since it is a 90% cure rate, then it's pretty high. So this is my presentation, and these are my references. If y'all have any questions, please let me know.